But now we can start. Okay. That's very awkward to say that again. Anyway, I'm very excited to introduce Aakarsh Vermani. He's a second-year student studying EECS at Berkeley. He's broadly really interested in machine learning applied to biology, and he conducts research with Professor Yoon Song, working on machine learning applied to interesting problems in genomics. And I'm particularly excited about the talk he's put together today because I think he's spent a lot of time into sort of figuring out how to condense these three different bodies of work, sort of RFDiffusion for protein design and backbone generation, ProteinMPNN for inverse folding, and AlphaFold for structure prediction, sort of how they relate to each other, and in the broader context of protein engineering campaigns. So with that, we'll give them a round of applause.
All right. Thank you so much, Sayon, and thank you everyone for being here. Let's get started. So yeah, today I'm basically just going to be talking about some of the seminal advancements in machine learning-based protein engineering from the last couple of years. And as you'll see, these have not only had major implications for biological and medical research, but were huge advancements in machine learning as well. So first, let's talk about why we really care about proteins. You know, they're actually responsible for more than just building muscle in the gym. They're a fundamental functional unit of basically all living systems, and they carry out every cellular process from transporting nutrients to replicating DNA. And so getting a better understanding of proteins gives us a better understanding of biology as a whole. They're also pretty important in many disease mechanisms. So an example is like Alzheimer's is a disease that affects the brain, and what happens is there's a buildup of misfolded proteins in brain cells. And so if we can understand, you know, what causes that to happen, we can potentially come up with a cure. Proteins are also involved in like a lot of therapeutics. A very popular example is like CRISPR, for example, where the main functional unit is the Cas9 protein that actually makes the edits in DNA. So that's another use case for proteins. And another one that is maybe not as talked about is like chemical engineering, because proteins often catalyze reactions, so they can be used to catalyze industrial reactions. And they can also be used in materials engineering. So a lot of materials that we might not think of are actually made of proteins, something like silk, for example. So we could potentially make materials artificially with proteins as well. So I have a feeling there might be like a fairly high variance in terms of biology knowledge. And in fact, why don't we get a show of hands? Like, who is like a biologist by education or works on biology problems? You want to raise your hand? And then, okay, well, maybe about half of the people here. And then how about who has like a more of a computer science background? Okay, that's probably like a little more than half. So I think this will probably be good for everybody. So we're kind of all on the same page. So the building blocks of proteins are called amino acids, and there are 20 unique ones. But they all have this general structure. So on the top part on the left, we have something called an amine group. And on the right, we have a carboxylic acid group. And they're both connected to what's known as an alpha carbon. And so that part is the same for all amino acids. What's unique is what's called the side chain, represented by the R. And these are basically extensions of the molecule that are and they vary in size and chemical properties, and they're what make each amino acid unique. And then, so these amino acids combine when the amine group of one amino acid binds to the carboxylic acid group of another amino acid, creating what's known as a peptide bond. And when you have this several times happening, when this happens several times, you create what's called a polypeptide chain. And it's these chains that actually fold to create proteins. And oftentimes a protein actually consists of multiple chains folded together. Some other like terminology, when you hear the term protein backbone, that refers to just the parts of the amino acid that aren't the side chain. And also sometimes amino acids are called residues when they're in this format, but it's somewhat interchangeable in practice. So let's say we have some protein involved in a disease mechanism, and we want to design another protein to be able to chemically bind to that target and inhibit its function. How would we even begin to start doing that, right? So this is a pipeline that's used by the Baker Lab at University of Washington. They're one of the global leaders in computational protein design. And so actually in this pipeline, they end up using all three of the models we'll talk about today. So they kind of start by trying to design a backbone or a general structure that might contain like a structural motif that would bind to this protein target. And then from that structure, they'd have to design the sequence that would eventually form fold to form that backbone. But then to predict how closely the folded protein will actually align with their original design, they have to somehow predict the structure from this new sequence that they've created. And then finally, none of that really matters unless it's all validated in a wet lab, because we need real experimental results to back this up. So we're actually going to start a little bit backwards because I want to go in order chronologically. And as you'll end up seeing, the first model we're going to talk about, AlphaFold, is actually pretty essential to understanding the other models. And they probably wouldn't have been possible without this first breakthrough. So AlphaFold was created to solve the structure prediction problem, which is to be able to determine the three-dimensional structure of a protein based on a one-dimensional amino acid sequence. And in the summer of 2020, Google DeepMind unveiled their second version of this model, and it did exactly that. And it did it pretty well, as you'll soon see. And the date I put here is 2021, actually, and that's just because that's when they published the paper and actually released all their code and predictions, which is what allowed many of the subsequent benefits. And so why do we care about this structure prediction problem? So it's a well-known fact that a protein structure is what determines its function. So the structure can actually tell us how the protein works. But the problem is that if we look at the available structure data we have, there's not that many. So the main resource protein data bank has only about 200,000 experimentally determined structures, which might sound like a lot, but if we want to do machine learning, we need a lot more. If you think about like LLMs like ChatGPT, they train on data in levels of petabytes. So really, there's a huge distance in terms of available data. It's like orders of magnitude there. This is also a problem we've been working on for a while. So since all the way back in 1994, this contest called Critical Assessment of Structure Prediction was started to basically test how good models are at coming up with the structure of a protein based on its sequence. And spoiler alert, but AlphaFold is kind of like the Michael Phelps of this thing. So, and then lastly, as we saw earlier in the protein engineering pipeline, AlphaFold can kind of take on the role of like a computational filter to see how close de novo sequences are to their design structures. I also want to bring up this slide. It's a pretty cool graph about the kind of sequencing revolution that's happened, that's made access to sequence data super accessible. But there hasn't been a similar speed up in like structure data. So logically, we'd want to take advantage of this kind of revolution and use it to get more structured data. So now I'm going to do a bit of a history lesson, actually. It might feel like we're back in high school, first biochem, now history. But so research from actually decades ago shows that the primary sequence alone of a protein can determine its final 3D structure, at least for globular proteins in like standard environments. It's an idea called Anfinsen's dogma. So theoretically, we should be able to just like figure out the structure from the sequence alone and using nothing else. The problem is that the like modern physics-based methods just can't scale to proteins because it's related to something called Levinthal's paradox, which is basically the idea that there's like an unfathomably large number of conformations that these proteins can fold into. And a protein can do it in a few seconds, but we haven't figured out how to do that computationally. So we're not really the giga-chads that Anfinsen would have liked us to be, but today our modern techniques kind of rely on external information, oftentimes related to evolution. So one of these is like homologous structures. So these are basically structures that are similar and are like evolutionarily related to the sequences that we might want to predict the structure of. And another type of data we rely on is called multiple sequence alignment. So multiple sequence alignment is kind of what it sounds. You basically align related protein sequences, starting with the sequence of interest at the top, And then, so as you can see here, we have protein sequences where each of these represents a amino acid. And in each subsequent row is also another sequence. And typically they get more and more like distantly related the further you go down. And in this example, the related proteins are analogs from different species, which is often how the MSA is formatted. In this case, we have human, the chimp to gorilla to orangutan. So why would this be useful in determining protein structure? So it turns out there's actually a lot of useful information encoded in here. So for example, let's take a look at these blue amino acids, right? So we start with one amino acid, and then it keeps changing in each subsequent sequence. And what that can kind of like hint to us is that maybe, you know, this protein, it seems like it's changing a lot. Maybe it's not actually that important to the structure of the protein. This is something called like a tolerant position, and it can give us some insight into how the final protein structure might look like. Another thing you'll see is have these red amino acids, and you can see that this V changes to a T over here, but at the same time, a R changes to K, and it's kind of continued throughout. What this could hint to us is that, so I'll use this diagram as an example. Say this is like the V that's changing to the T. This kind of implies that maybe the reason that the R changed to a K is because they were interacting closely, and by this one changing, the other one had to also change to make sure the structure was still stable. This is an idea called coevolution, and it's another kind of signal that we can kind of pick up from a multiple sequence alignment. This is just kind of a contrived example, but it's signals like this that help models like AlphaFold make really good predictions. Okay, so now that was a lot of background information, but now we can actually start talking about the meat and bones of the thing, which is this architecture of AlphaFold 2. And we're going to start with the inputs. So as you might expect, we start with a protein sequence, and then there are kind of two different searches. First, we start with a genetic database search, and this is what helps us construct that multiple sequence alignment. And then the multiple sequence alignment goes from this two-dimensional matrix to a 3D matrix, where the dimensions are like the number of species, the number of residues. Oh, sorry. And then there's like this depth of length C. And so the reason we put it in this 3D representation, kind of the intuition behind this is actually can kind of come from natural language processing, where words are often represented as vectors. And the idea is that like a higher dimensional representation will be more informative than just some scalar value. And so the hope is that the model will be able to embed meaning into this vector of length C that now represents an amino acid. Meanwhile, they also create an R by R matrix, and that compares each residue. So it basically just compares each residue to each other residue in the sequence. So this is a more of a close-up into this. And so we can see how this matrix essentially represents the amino acids in the sequence as a graph. It's kind of like an adjacency matrix, if you're familiar with that. So for example, position at like ij will represent the edge in the graph going from I to j. And then just like the MSA representation, we're using a vector of length C to ideally capture more information than just a scalar. And the last thing to note here is there's the structured database search. And the idea is to just try to see if similar structures exist to try to initialize the pair representation matrix if possible. And then before we talk about the next portion of the model, which is the evoformer, there's some key intuition I want to make sure everyone develops about a machine learning concept that's been pretty instrumental to the ML boom we've seen in recent years. So it's this idea called self-attention. And I'm not really going to talk about any of the math or the implementation details, but just the basic intuition of what it means. So when we have data in a sequence as represented by these nodes, they could be anything from like words to amino acids. In this case, I made them amino acids. It's often useful to be able to capture how important individual items are in relation to one another. And so that way the model can be selective with its attention and in deciding which inputs to care more about. So in this example, we can see that the model can tell that maybe K will provide a lot more useful information about P, but D not so much. And this concept of self-attention is largely what's made transformers like GPT so successful, because now we can actually capture long-range dependencies. And it's especially important with proteins because if you think about it, you could have amino acids from opposite ends of a chain interacting with each other. So kind of capturing these long-range dependencies is pretty important. So now let's talk about the evoformer portion of the AlphaFold architecture, which as the name kind of suggests, it's a modified transformer. And so first we have two self-attention modules to capture those dependencies I just showed. So there are two models because self-attention is applied both row-wise and column-wise to the MSA matrix. And this idea is actually not unique to the evoformer. It's called axial attention and was first introduced in computer vision, I believe. And it's important because we want information encoded across the individual sequences or the S dimension, so along the column, because that's the information as we showed through that exercise earlier that could gather information from the columns of the MSA. But we also want information about the relationship between amino acids within the same sequence. And that's going to be along the rows or the R dimension, because that's ultimately what we're trying to predict. The thing is that MSA doesn't actually give us that much new information about these rows alone. So that's why, as you can see by this arrow, there's actually a bias term that's passed from the pair representation into the calculation for this row-wise attention. And it makes sense because the pairwise matrix is exactly what's meant to encode that relationship between different residues in a sequence. But it's not, the pairwise representation isn't that useful to the column-wise because it's not really representing different sequences. So that's why we don't really have the same bias term added over here. And then continuing through the architecture, we have this transition that's kind of just like a vanilla two-layer, multi-layer perceptron. And then the pair representation is updated through an outer product mean from the MSA. And I'm not really going to go into the math details, are not that important, but you can basically think of this as a way to update the pair representation with some kind of information from the MSA representation. And then the next several modules here are applied to the pair matrix. And the idea is to make sure that they are like mathematically consistent. And so what do I mean by that? So I have another like kind of contrived example here. So let's say we have this really small pair representation matrix that let's say we only have 3 amino acids and that each entry in the matrix represents the distance between these amino acids. And this is not how the pair representation in AlphaFold works in reality. This is just like a simplified version, but we can assume that the encoding would somehow include this information. So if we see that I and J are 2 units away and J and K are 3 units away, we also have that I and K are 7 units away. And so hopefully if you have good mathematical or spatial reasoning, you can see why that might be a problem. And it's because it violates this idea of the triangle inequality. In essence, one leg of a triangle can't be larger than the sum of the other 2, or else the points can't all connect. And that's what would happen in the pair matrix I just showed. So there need to be a way to kind of make sure this doesn't happen. And so there's 2 different methods that they kind of employ. The first solution is pretty simple, actually. They basically just consider every possible third point of the triangle. So if we're considering the vector at entry ij in the pair representation, then we're basically applying a multiplicative update of the dot product of these 2 other entries at ik and jk, which represent the edges of the triangle for every possible k. And this is done for both outgoing and incoming edges, which I put those in quotes because just like kind of a reminder that this graph structure isn't like explicitly coded in the model, or at least this portion. It's kind of just like our interpretation of the representation within the pair matrix of the model. And then the second thing they do for the same reason is another modified form of self-attention. So for each edge ij in the pair representation, they basically compute attention values for each edge that's like coming from point I to each of the other possible k values to get a sense of how important the other edges are. And to help with calculating that value, they include a bias term from what would complete the triangle from JK, which is kind of similar to how the hair matrix kind of updated the row-wise self-attention earlier in the Evo former. And again, they do this both for outgoing and incoming edges. I would also like to point out that from a time complexity standpoint, these triangle algorithms are pretty costly, as they involve considering every possible triangle that can be made in the protein. So that's like a O of R cubed algorithm. But yeah, that's the Evo former. There's kind of a lot of details there, but the main takeaways I would get from this are that the MSA and the pair representation matrices are actually like learning from each other, which is a really cool idea. There's this usage of axial attention because both the rows and the columns of these matrices are important. And then also the way the triangle inequality is dealt with, it's not like some hard-coded thing, but rather it's kind of encouraged by providing this information about like potential third points in a triangle. And in that sense, they're probably also helping make the model better just by allowing it to consider more information. And then I also want to point out that this isn't just happening once, it's actually happening 48 times, which is pretty crazy. And so the last part of this architecture is the structure module. So we see that our inputs to the structure module are the, so that single sequence representation, that's basically just the top layer of the MSA matrix. And the one that, that's the one that actually represents the sequence that we're looking at, and hopefully would have like a very rich and meaningful embedding by now. And then we're also passing in the pair matrix, which should have useful spatial information. And then the backbone frames, which is how the model actually represents amino acids in 3D space. So let's look into what these are. So again, let me reintroduce you guys to our good friendly amino acid. So AlphaFold actually takes advantage of the fact that these bonds are fairly rigid. And so it kind of represents this amino acid as one triangle. Note that we can't say the same thing about this angle, this bond angle specifically, but we'll come back later. So this representation is called a backbone frame. And the triangle is basically defined by two features. So one of them is the location of this corner of the triangle, which represents the alpha carbon. And its location is just a three-dimensional vector. And the other feature is the orientation of this triangle. So basically, in terms of orientation, we can kind of think of each of these triangles as having its own local reference frame. And we can represent the orientation of the triangle as some rotation matrix that takes us from a global reference frame to a local reference frame. So this matrix is combined with the location of the alpha carbon, and that's all we really need to represent any amino acid in 3D space in this model. So another interesting aspect of the backbone frame representation is that during when the model runs, there's actually no requirement that the residues need to be connected. So they kind of call this a residue gas. But at the same time, there's a term in the loss function that penalizes the model if the prediction has residues that aren't connected to one another. And they also do like an energy minimization of each prediction. So that's usually enough to ensure that the residues are properly connected. And kind of thinking back to like the two schools of thought, that's probably the only real physics-based feature of AlphaFold 2. So now that we understand how amino acids are spatially encoded in the model, let's talk about what might be one of the most exciting results from this paper from a machine learning perspective. It's this very desirable property called like SA3 equivariance. So the idea behind this that's desirable in machine learning is we want the model to treat objects the same, even if they've been somehow globally rotated or translated. And in the case of protein structure, that's pretty important because protein structure prediction, that's important because we don't want the model to think that a protein is fundamentally different just because it's been like rotated or shifted a little bit in space. So AlphaFold is able to implement a form of attention on 3D points that satisfies this condition. It's called invariant point attention. And so to best see why this works, let's actually take a look at how the attention weights are calculated. There's a lot of stuff going on here, but I'll point you to the things that actually matter for developing intuition. So The attention weight capturing the relationship between the different residues I and J from a 1D sequence based on the input sequence is right here. And that's kind of how a normal vanilla attention term will look like. But then there's also this 2D bias that's pretty similar again to how the pair matrix updated the row-wise attention in the EVO former. And then finally is this 3D term, which you can basically see is kind of in the form of a Euclidean distance calculation, which implies it kind of captures the distance between the 2 points. And they've applied these TI, TJs, which represent those local reference frames as well as the location of the alpha carbon. So it's clear that this point attention is capturing all the important information we have from all these 3 different levels. But why is it invariant? And so there's actually a pretty simple proof that they use in the paper. So let's assume that you just applied some kind of global Euclidean transformation called T global to the whole protein. So the new expression for that term would be this. But then what we can see is we can essentially just factor out the T global. And then since it's going to be an orthonormal matrix, it doesn't actually end up affecting our L2 norm. And so our attention calculation is still the same. And then similarly, when we actually apply all these attention weights and across all the other residues, and then we get some output, the calculation is indifferent to global transformation. So if we were to again include some kind of global transformation here, they end up canceling out nicely, and we're left with a expression that is not dependent on any global transformation. So just like that, we can see that the invariant point attention calculation is indifferent to global transformations, which is super useful. So the outputs of the IPA module are then passed into a backbone update algorithm that will update the positions and orientations of these backbone frames. And the final step of the structure module is to predict what are called chi angles. And these are what determine the orientation of the residue side chains, which, as I mentioned earlier, can't actually just be treated as rigid like the other bonds in the amino acid backbone can. And this actually ends up being a much simpler task, and it's just performed per residue through a small neural network. And yeah, so that wraps up the architecture of AlphaFold 2. Some other key points to notice are this recycling that happens where the model runs completely and then feeds back its predictions into the start 3 times. The other important thing about this architecture is that it's completely end-to-end differentiable, which means that there's back propagation or feedback throughout the whole model, which is something that the earlier version of AlphaFold actually didn't do. There, the 2D reasoning was distinct from the 3D reasoning, and so it ended up losing a lot of performance because of that. It's also probably good to mention that the loss function is a weighted sum of a few different metrics. The main one is a frame-aligned point error, which basically calculates how far off predicted atom positions are from the ground truth structure, with various alignments to remain agnostic to any global translations or rotations. I also wanted to talk about some of the more interesting results from the ablations, basically chaining aspects of the model to see how it affects performance. I think it's interesting that having no templates, as you see here, doesn't actually affect the model performance that much, which I think is pretty crazy. The other features that made a pretty big difference, though, include triangle attention. There's recycling, the end-to-end differentiability, and invariant point attention. You can see here no IPA and no recycling makes it a really big difference on the model performance. So to sum up some of the key highlights of AlphaFold 2 performance, so they did really great at CASP 14 in 2020, and they approached experimental accuracy in terms of structure determination, and they had a summed z-score of 244, and for reference, the second place was 90. In fact, they did so good at CASP 14, they didn't actually have to compete in CASP 15 to still kind of dominate. So I found this fun little word cloud of all the abstracts submitted to CASP 15, the next iteration in 2022, and you can see that one of the major words that was written was AlphaFold. And I think it appeared on average like 1.4 times per page in the abstracts of the CAS 15 entries. So even at the next competition, almost all the models were based on AlphaFold in some way. The ability also to provide a confidence score through something called predicted local distance difference test, which is like a kind of a score like per residue, has also unexpectedly helped us identify certain things like intrinsically disordered regions of proteins. And it's pretty useful for having like this kind of metric for AlphaFold's prediction for the reliability of its predictions. It's also been bootstrapped for a lot of other purposes, such as, for example, predicting protein-protein interactions that have been pretty successful. And perhaps most importantly, it's been used to develop like 1 million more accurate structures, of which approximately like 1/3 are very high confidence. So obviously, this is a huge leap from the 200,000 experimentally determined I mentioned earlier. And there's a protein structure database from AlphaFold that contains the structures of the whole proteome of humans and 20 model organisms. So that's like 360,000 structures in total. A few limitations though of this version of AlphaFold so it only really works on globular proteins and doesn't work as well with certain proteins that are embedded in like membranes, which have different environments. And it also can't really work on like protein complexes. Another issue with AlphaFold is that it's bad at dealing with de novo mutations. So if we like introduce a point mutation somewhere, it's not really good about reasoning physically about how the mutation would affect the structure. And that makes sense because as we saw, all of this kind of signal is coming from like evolutionary information. What that also means is that without like that homology information, like without that MSA information, it will perform significantly worse. Okay, so that was AlphaFold. I wanted to give people a chance for any questions or comments. Yeah, Terry.
I'm kind of missed the part where you said there were numbers that violated the same way.
Oh, yeah.
What were the numbers? I think I just know.
Yeah, so like I said, this was like a pretty contrived example. The idea is that we want the model to reason in a way that's like geometrically consistent. Because if it predicts that like I and J are really close and J and K are really close and interacting, but then it predicts that I and K are really far apart, there's no interaction. That doesn't really make sense, right? Like geometrically, we can't really have that happening. Does that make sense?
This is
Yeah. Awesome.
Could you like re-elaborate on like what some element of this matrix means again?
What of the matrix?
Like this pair representation matrix, like element ij, what would you say?
Yeah. So in this contrived example, I'm saying it represents like the distance between like those two residues. In reality, like this is not what the pair representation would look like. It would be some vector, but this information would likely somehow be encoded in it. Yeah.
Is there, are you aware of any work being done kind of at the frontier of incorporating new modes of experimental data into AlphaFold 2 and other similar models to kind of basically act as a form of reinforcement learning to update the model's kind of model structure predictions?
That's a really good question. I'm not personally aware actually, but that does sound like something that would be pretty useful, and I'm sure there's people who might be working on that. Yeah. Okay, so I'm just gonna get a quick drink of water and then we'll jump into the next model on the list. So going back to this protein engineering pipeline, we saw how AlphaFold could be used to validate generated sequences by making predictions from a sequence, and obviously do a lot more. But if we're engineering novel proteins, how do we even like get these sequences in the first place, given some kind of protein backbone? So that's where ProteinMPNN comes in. And so I decided to use the same graphic and then throw in that nice little arrow switch, because that's essentially what this is. This inverse folding problem is, instead of going from sequence to structure, it's trying to go from structure to sequence. And so as of its release in September 2022 by the David Baker Lab at the University of Washington that I mentioned earlier, it was the state of the art and what's called the inverse folding problem. And as I mentioned, it's just trying to do the reverse of AlphaFold. So let's kind of assess the state of computational design, protein design at this point in time. So AlphaFold was obviously a game changer, and many people consider the structure prediction problem like almost essentially solved, since they were able to, be on par with experimental techniques for determining protein structure. And any, further improvements would just kind of be incremental. But other aspects of the protein structure pipeline, namely structure generation and inverse folding, were kind of lagging behind. But AlphaFold was able to open the doors by introducing new deep learning techniques for reasoning about proteins, and then also providing a huge increase in structural training data. And then, as mentioned, like kind of providing this new method for benchmarking performance through its role as like a filter in that pipeline. So inverse folding is crucial because if we have a specific structure we want to create, it allows us to, it gives us the protein sequence that will actually fold to create that structure. And through some kind of structure prediction like AlphaFold or through wet lab experimentation, we can actually test out the design, which we wouldn't be able to do without a sequence. So that's really the key thing here, is that this allows for protein designs to become realizable. So the key idea behind ProteinMPNN is the MPNN part, or message passing neural network. In a nutshell, it's a neural network that takes in a graph in the form of nodes, edges, and their respective features. And a given node representation or embedding is then updated through an aggregation of the representations from the local neighborhood. So this paradigm is called message passing because the information is kind of like just being passed along the graph, thanks to this aggregation function. So note that with AlphaFold, they used one-dimensional self-attention because we wanted to capture long-range dependencies. Because you could, you could have residues at opposite ends of a chain interacting with each other. But when we're doing the reverse and we're trying to go from structure to sequence, we really only care about the local groups in 3D space, because a residue on the other side of a protein isn't really going to have much of an effect physically. So ProteinMPNN is actually heavily based on this baseline model from a 2019 paper, and it's actually pretty similar to there's a lot of similarities to like a standard transformer architecture. So the major differences are the model is taking in a graph as an input, considering edge embeddings and node embeddings. And through that, it's able to kind of encode a structure. And you can't really tell from this diagram, but a key thing to note here is that only the node embeddings are being updated and not the edge embeddings. So after the structure has been encoded, the decoder model will output its amino acid predictions in the form of probabilities. And the decoder works what's called autoregressively and using causal self-attention. So basically what it means is that it outputs these predictions sequentially, and considers sequence only for the positions for it. So if we start with, say, the first amino acid in the sequence, it will make that prediction based only on the structure because that's all it has. But then when we go to the second amino acid, then it will have its prediction already from the first one. So it'll be able to use that sequence information as well. The question?
Can you clarify how the nodes are getting updated then? And then you said the edges aren't getting updated. It's just the nodes. Could you clarify why?
Like why the edges aren't getting updated?
I mean, how do the nodes?
Yeah, the nodes or embeddings are getting updated through like back propagation. The edge embedding. So I bring this up because in this model they specifically don't update the edge embeddings, but in the actual ProteinMPNN, that's like kind of an update that they make. As for why they didn't do it in this paper, I'm honestly not too sure. So basically, what this causal attention means is that so the model will only look at sequence from the things that it's already predicted. So that's kind of represented by these colored arrows. So like the red ones, it's able to use both structure information and sequence information coming from those red arrows. But from these blue arrows, it only gets structure information because it hasn't made predictions about the sequence yet. And then the other important thing to note here is that in line with the message passing paradigm, self-attention is only calculated for the k nearest neighbors. So in this paper, it's 30. And so this makes the attention calculation actually significantly more efficient. So normally it would be O of n squared because you're comparing each residue to each residue, but now it becomes just O of nk. So this is how the graph is specifically encoded at the start in the original MPNN model. The edge embeddings are a combination of some structural embedding, which incorporates the distance between the nodes here, the direction difference between the nodes, as well as the differences in orientation. This O matrix is very similar to the R3 that we saw from AlphaFold. As each residue is basically modeled as a point that has its own like local orientation or like frame of reference. The edge embeddings also include a positional element, which is defined as a sinusoidal function of I minus j, where I and j are like the position number of the amino acid in the sequence. And then the node embeddings are based on these angles of the protein backbone. But if you remember from AlphaFold, we didn't actually take these into account and just kind of assumed it's a rigid triangle. And as we'll see in a bit, that approximation is actually pretty valid because in the final ProteinMPNN, they actually just kind of get rid of these initial node features. So this original MPNN for inverse folding did pretty decent. It averaged about 40% sequence recovery on test sets, and that might not sound very great, but it was very good for the time, and it was beating out the Rosetta suite that was made by the Baker lab. So they obviously had to come up with a way to take back the throne, and what better way than to just make improvements on what your competitors design, right? So right off the bat, you'll notice that we have this nice little like Christmas color scheme going to highlight that ProteinMPNN can actually deal with multiple chains, which is a huge benefit of the model. And I'll explain soon some of the features that give it that cool ability. So let's first look at the inputs. So edge embeddings are calculated for the nearest neighbors in the same way as the baseline MPNN. The major difference is that they also include, here in red, they also include the distances between the rest of the atoms in the amino acids, not just looking at the alpha carbon like the original MPNN was. And so as you'll see in a bit, this ends up helping a lot. Also, the positional edge embedding is slightly different, so they don't use a sinusoidal function based on I minus j. They basically just take that raw difference, of which there's like a maximum of 32. And that's actually exactly how AlphaFold does it as well. And then it also indicates if the residue is from the same or from a different chain, although in the supplementary information, the authors actually indicate that the position encoding isn't that important based on ablation studies. So the input for the node features, as I mentioned earlier, is actually just zeros, since the information gathered from the edge inputs ends up being all that really matters. And the main architecture is pretty much the same as the original MPNN, even though this diagram does look a little bit different. The key difference in the encoder is that both the edge and the node representations are being updated, as you can see through these cycles, as opposed to just the nodes in the other MPNN model. And then in the decoder, the main difference is that instead of the sequence being predicted in some kind of sequential order, like in a fixed sequential order, it's actually decoded in a completely random order. And so the reason for this is they were.
In the previous slide, what is the 3x that you have?
Oh, yeah, so that just represents this is like a 3-layer model, yeah. So the Baker lab was trying to be very practical in their design of protein MPNN. And a common desire in protein engineering is to design a protein around like a specific fixed motif. So meaning there might be certain sections of the sequence that are predetermined. And so in the fixed decoding model, if the motif is like in the middle of the protein, the decoder won't be able to use the valuable information that comes from that fixed section of the sequence. So if you look in this diagram, if we're just going in order from left to right, these first two predictions are not going to be able to use the sequence context that these two positions provide, which means it's just losing out on valuable information. But during ProteinMPNN's order agnostic autoregression by randomizing the decoder order during training, it means that you can kind of arbitrarily decide which order to go in. So basically, you could decide to start with the parts of the sequence that have been determined and then go from there. And so now you see that each subsequent prediction is also using the sequence context, and it's making use of that valuable information. So not only does this let the model bake in those fixed motifs, but it also ends up just increasing performance in general. And it kind of makes sense intuitively because if you think about the relationship between amino acids, it's not necessarily ordered from left to right as words might be, as you might see in like language, for example. So it does kind of make sense why this works. So let's see how those updates, yeah.
Sorry, in the previous slide, did you say the like autoregressive context number is like randomized? So I mean,
you can choose the order in which the sequences, the predictions are made arbitrarily.
okay, is there like a standard that they like set to like okay, randomized, like, or was it just fully randomized? So just the G is fixed, that's 1, 2, and then after that the boost was like it could be 3, 4, 3, 5, 4, or like.
Oh yeah, that's a good question. I'm not sure. I think this might just be like this small example that they gave to show that it can be random. I'm not sure if like when they're actually making predictions, they like after doing this, they then go in some order, or it's like fully random. That's a good question. I think that
They just randomize everything that's not fixed. Like all the remaining entities, we just like random, just like.
yeah, okay. Thanks, Sam. See, so yeah, let's see how those updates to the baseline model did. And so right off the bat, just by including the other atom distances in the edge inputs, test accuracy actually jumped by like 8% here on a PDB test set, which is pretty significant. And then when you throw in the fact that they're also updating the edge representations in the encoder, that as well as this random decoding order, then they increase accuracy by roughly 10% using just these three simple but clever adjustments. And then also note here that perplexity is just a common metric used in sequence modeling, and that in this case, lower is just means the model is performing better. So another key feature that ProteinMPNN authors added was this idea of like noise during training. So on the test set I was pointing to, you can see that the accuracy actually decreases in this situation where some noise is added. But this is for structures that are determined with a high accuracy through experimental methods like X-ray crystallography. But when you run on AlphaFold structures, the noise ends up actually helping you because the backbones aren't going to be like quite as accurate. So the main use case of ProteinMPNN, if we think about it in this the context of this like computational protein design pipeline, is to realize these computationally designed backbones. So while it may reduce the PDB accuracy, it might actually lead to better accuracy on a task that we care more about from a protein design perspective. And then the last,
Why do you think it's not like always PDB accuracy of AlphaFold accuracy of?
oh, like why is it? Yeah, so I think it has to do with the fact that AlphaFold probably just varies more in its accuracy, and so adding kind of accounting for that with by adding noise can help the model generalize better. Whereas PDB is going to be a lot more in general for the X-ray crystallography determined structures, it's going to end up being a bit more accurate. Yeah. So the last interesting feature of ProteinMPNN I want to talk about is this idea of like coupling residues across chains. So it's often desirable for design proteins to exhibit some degree of symmetry. And so let's say that we have protein backbones consisting of what are supposed to be 3 symmetric chains, or what's called a homotrimer. So what the model does is it predicts an unnormalized probability for each of the chains. So in this example, so we have like A3, B3, and C3. And then it will basically average out this probability across the 3 positions, and then outputting like the argmax, or basically the residue with the highest probability, to each of the 3 positions. And this actually ends up working out really well. In fact, the class of proteins with the highest average sequence recovery accuracy is like proteins that are designed with this symmetry of mind, with this chain coupling. Yeah. So, yeah, yeah.
I'm sorry, the symmetry part, how do we know that it's protein is gonna take the structure of the 3 triangle, or like 3, or like 4, whatever?
Oh, like how do we know? So I think it's assumed that you know that like you're designing this protein as with the goal of it being 3 symmetric chains. Yeah, yeah. So it's assumed that like this is something you specifically want to make. Yeah. Okay, so some of the highlights of ProteinMPNN's performance. So at the time it achieved state-of-the-art accuracy, and as of today, that's not necessarily true, but I'll talk a little bit more about that at the end. And perhaps more importantly, though, it was the first model that could actually accurately deal with multi-chain proteins, or multimers, which is a big deal for protein design, since these multimeric proteins are often more useful. And then another interesting result from ProteinMPNN is that they found that on average, the AlphaFold productions using ProteinMPNN generated sequences actually more accurately matched the actual structure than AlphaFold prediction on native structures that didn't have MSA information. That's a pretty big like caveat, that didn't have MSA information, because that tends to make AlphaFold a little bit less accurate. But this does imply that ProteinMPNN sequences result in pretty highly stable and favorable structures. And there are extensions to this model showing that could actually be really useful for increasing stability of designed proteins. And then lastly, unlike most other inverse folding algorithms, there was extensive experimental validation, which is always good to corroborate computational methods. So specifically, they tried expressing 96 sequence predictions in E. coli, and about 50 were produced with high concentrations and exhibited the desired protein structure, which aligns really well with what the sequence recovery metrics said. But then some important limitations as well. The accuracy isn't that great. If you like the sequence recovery accuracy, like I said, is in the realm of like 50%, which isn't that great. But I suppose you could also argue that it's a good thing, because that means there's a lot of room for improvement and some exciting new work. Also, if you look more into where accuracy is failing, there's a clear trend in like the sequence recovery versus the average distance of an amino acid from its 8 closest neighbors. So for amino acids in the center of the protein, where there's often much higher density of residues, you get like really high accuracy metrics of like 85, 95%. But then when you get to more sparse areas of the protein, where the average distance between its closest neighbors is a lot larger, then the accuracy significantly drops. So clearly there's like this dependence on having neighbors close by, which is limiting kind of the overall accuracy of the model. Overall, though, is definitely a very important leap in inverse folding and deep learning based protein design in general.
As a clarification, is the accuracy kind of ground truth here being normalized against X-ray crystallography data, or what is like the ground truth experimental data?
The ground truth experimental data is like the actual native sequence of the protein.
Oh, I see. So you're trying to predict the native sequence from a predicted, or like a experimentally measured structure.
Yeah, from a structure, exactly.
Okay,
yeah.
and the structure is measured from like extracrystallography, or is it
Yeah.
IOM or something? Yeah.
Yeah.
Okay.
So even though it's metric for 8 closest neighbors, for every calculation, they always looked at the 32 neighbors.
Yeah, this specific one though, it just looks at 8 closest neighbors. Yeah. Yeah.
Do you have thoughts on like how to solve that problem of density? Like if you start relying on having closer distance?
Yeah, no, that's a really good question. I don't think I have any specific ideas in mind. But yeah, I mean, it makes sense because the kind of the main signal you're getting is like looking at the interaction of neighboring residues. So it seems hard to, kind of get over that hump, but maybe a potential solution would be trying to integrate some other source of information.
Are those high distance clusters correspondent with the low confidence prediction clusters in AlphaFold?
I'm not sure actually. That would be something that would be really interesting to check. Yeah. All right. Are there any other questions? And if not, I'll take another drink of water and then we'll jump into the final model. All right, so now we're heading back to our handy dandy protein engineering pipeline, and we're onto the last model, which also actually happens to be the first step in the process, which is backbone generation. So the model I'll be talking about for de novo backbone design is RFDiffusion, and this is the newest of the models we talked about, and it was developed by the same creators as ProteinMPNN, the Baker Lab. And the respective paper was published last summer. Although it's also worth noting, this was a pretty massive collaboration. It also included several co-authors from Columbia, MIT, Harvard, Cambridge, and others. So, okay, let's do another kind of recap of the state of the computational protein design space. So we've had big improvements in inverse folding and structure prediction by now, but we want a good way of designing backbones in the first place. And why? Well, first of all, the set of possible proteins is unfathomably large, as I mentioned. And so nature has really only explored like a kind of a tiny fraction of these. And so there's bound to be highly useful proteins out there that have just never naturally folded. The goal is, with reasonable accuracy, can we design proteins that obey the laws of physics and biochemistry so they actually fold? And then can we also make them novel? So we aren't just, kind of reinventing the wheel and redesigning native proteins. And can we make them display the traits that we want so they can be used for a variety of purposes, like some of the purposes I mentioned at the very beginning of the talk. So at this point in time, a lot of the leading methods involved something like inducing hallucinations of AlphaFold and other structure prediction models. But these were pretty slow and not great in accuracy. So RFDiffusion kind of swooped in with to make some pretty big improvements in a new direction. As you've probably noticed by now, like a lot of these advancements in bio-ML pretty closely follow advancements in other ML disciplines, things like NLP, like natural language processing, and computer vision. So RFDiffusion is not an exception to that, as it makes use of a pretty popular type of model called a DDPM, which is also the basis for a lot of popular image generation platforms, things like DALL-E, that I'm sure a lot of you have heard of. So denoising diffusion probabilistic models, or DDPMs, how do they work? So the general idea is that we start with some kind of ground truth, say like this S, which we call X naught. So over a series of T time steps, we iteratively add Gaussian noise until we get essentially completely random noise at this time step X capital T. And so this is called the forward diffusion process. And then it's in the reverse diffusion process that we want to be able to predict previous time steps based on the current noise one. And we do this by training a neural network to learn how to do noise until eventually we have a sense of the probability distribution that describes X naught. and from that distribution sample an x naught hat, like an estimation that hopefully captures the characteristic features of our training data. And in the case of proteins, it's going to be a very similar idea, but now we're dealing with amino acids, and we're trying to make a protein, not an S. Okay, so now that we have a basic understanding of a DDPM, let's get into RFDiffusion. So just like AlphaFold, RFDiffusion treats each residue as a rigid triangle defined by a rotation matrix and the location of the alpha carbon. And so during forward diffusion, the protein representation is noised by adding 3D Gaussian noise to the alpha carbon location and noising the rotation matrix with Brownian motion or random motion that basically keeps the matrix in what's called SO3. So that basically ensures that the rotation matrix stays a pure rotation matrix and won't represent some kind of like scaling or translation. And so the idea is that by the final time step, the alpha carbon locations will follow a Gaussian distribution, and then the rotations will be something completely uniformly random. So the reverse process or denoising is probably the more exciting part of the diffusion model. And the main idea here is that the denoising model is actually a structure prediction architecture itself. So in particular, the creators of RFDiffusion used RosettaFold, which is basically the Baker Labs version of AlphaFold. It's using a very similar framework, and I think it achieves like slightly worse accuracy, but you also have the added benefit of being able to work with multiple chains. And you can see that kind of like AlphaFold, but more explicitly, RosettaFold takes in 3 levels of input. So there's a one-dimensional sequence, some 2D pair representation that's based on like previous homologous structures, and then a 3D frame representation. And so during this reverse leg of RFDiffusion, they're essentially training RosettaFold to be able to denoise random coordinates and sample from the probability distribution that describes valid proteins. So they pass in the same inputs, except now the input sequence is masked because we don't actually have that prior. And our 3D input is just going to be those random diffused coordinates. And then they also pass in something here to this this homologous template input that I will explain very soon. But basically, I think a really key idea here is this ablation that the authors did that showed that how RFDiffusion performs when RosettaFold is pre-trained versus when it's not pre-trained. I think it's pretty remarkable that when RosettaFold is not pre-trained, there's almost no difference between literal random noise and the predictions from the model. So what this basically tells us is that the major inductive bias of RFDiffusion is actually coming through this structure prediction model, showing again that this kind of advancement in backbone design wouldn't really be possible without those initial advancements in structure prediction. And just to clarify the metrics being used here, what they do is they like design a backbone, and then they get the sequence with ProteinMPNN, and then they get the structure from that using AlphaFold. And so kind of the idea here is that AlphaFold has implicitly learned a lot of the rules of protein folding. So the more similar the RFDiffusion generated backbone is to the AlphaFold design, the more likely it is that this protein can actually be made in real life. And so the first plot is showing the root mean square distance, where lower is better. And then the second plot is showing this kind of like confidence score from AlphaFold, where again, lower is better. And then they've also split the graph into 2 halves, unconditional and functional site scaffolding, or like motif scaffolding, which I'll explain in a little bit. So one of the other techniques that RFDiffusion used that ended up making a pretty big difference was self-conditioning. And basically the way this works is, so normally what happens is that at some time step t, you make your prediction, your x naught 0, and then you perform this kind of interpolation of your current state xt and your prediction of what the final protein is going to look like. And then you add a little bit of noise, and then that's going to be your prediction for the next step. So you do this iteratively to get to the next step. What self-conditioning does is it takes advantage of this X naught hat prediction that in a normal diffusion process just gets thrown away. It takes advantage of it and passes it in through that second input, specifically the homologous template input. I think it's right here. And so what this does is allows the model to kind of learn from its own previous predictions. And it's actually a very similar idea to recycling from AlphaFold. And basically by giving it feedback from its previous iterations, it's actually able to learn more effectively. And as we can see again from these graphs comparing without self-conditioning and with self-conditioning, that it ends up making a huge difference, not only like leading to better performance on average, but also kind of definitely lowering the variance of the results that we get. And so as we just saw, comparison to AlphaFold kind of computationally validated that RFDiffusion designs are legitimate proteins. But the second criteria we also wanted to look at is that proteins should be novel. Because again, there's no point in just like reinventing proteins that already exist, right? So this graph shows that the highest TM score with all PDB proteins are of RFDiffusion generated backbones. And the TM score can kind of roughly be interpreted as like a percentage similarity since it's on a scale of 0 to 1. So we can see that for proteins that are 100 amino acids long, the median highest similarity to an existing protein is only about 70%, showing that the model isn't just like, overfitting to training data. And as the length of the sequence gets a lot longer, that similarity goes down significantly. And that kind of makes sense intuitively because as the sequence gets longer, we'd also kind of have a larger space of possible configurations. So going back to like kind of those three goals that I mentioned with protein backbone design, the third important thing that we want is to be able to create structures that actually satisfy a specific criteria that we have. So RFDiffusion is a pretty flexible framework that allows for a lot of forms of this type of like conditional generation. And so one example is by allowing for symmetric designs. And a property that's pretty useful for protein design is symmetry, as I mentioned earlier. It generally makes proteins more stable, and in certain cases, they can be better binders. And so basically symmetry is enforced in RFDiffusion through a fairly simple process. Basically during the denoising steps, at each step you apply the symmetry operators for the type of symmetry that you want. So if you want three-way rotational symmetry, for example, you can just kind of take a third of your point cloud and then rotate it around to like kind of recreate it and make all three sections symmetrical in that way. And then you just kind of repeat this process throughout all the steps of reverse diffusion until you're left with a final result that is symmetric. Another criteria we might want to invoke in our design proteins is like specific types of folds or like secondary structures. So the design on the left represents helices arranged in a type of fold called a Tim barrel. And proteins with this feature often make great enzymes, meaning they can catalyze chemical reactions, so they're pretty useful. And in the RFDiffusion paper, they actually explain how they're able to easily design an adjacency matrix that represents this type of fold, this like block adjacency matrix, as well as encode for the secondary structure through the one-dimensional sequence, indicating like basically what structure feature each residue should belong to. They then pass in a combination of these two to that same 2D input template. So this same place where like the self-conditioning template goes in order to get the desired output. And so I have an example of some experiments they ran on this specific Tim barrel example, and they show that the designed protein had that same structure as a natural occurring protein while still having a overall unique structure. And then these plots, I'm not really going to go into detail about what they represent, but you can interpret them as basically saying that when they were able to be created in E. coli with pretty high concentrations and actually be expressed and created, And then the bottom plot shows that the protein is thermostable through a wide range of temperatures, which is obviously a good property. And so a little over 41% of the Tim barrel designs matched desired criteria. And that number is around 54% for another structure feature called NTF2, which is really efficient as far as protein design goes in terms of experimental validation. Okay, so functional motif scaffolding is like pretty similar to the previous concept. Instead of dealing with general secondary structure elements, we want to include motifs that serve a specific function. Performing this task with RFDiffusion is, again, like actually pretty simple. When you start the denoising process, you pass in fixed 3D coordinates of the amino acids of the motif you want to include. And then you basically just let the model update the surrounding residues. And so to test the performance of RFDiffusion on this task, they created a benchmark of like 25 different functional motif scaffolding tasks that were pretty common in the literature. And as you can see, RFDiffusion, which is represented by both the pink circle and the X, those just kind of represent different hyperparameters. It basically across the board is mostly the highest performer compared to like other techniques. Yeah, basically outperformed the other methods. And so what they did here is they generated 100 designs, and RFDiffusion was able to make valid designs experimentally for 23 of the 25 tasks. And the next two highest models that were some form of like hallucination and in painting were only able to get like 19 and 15 of the tasks. So how am I doing on?
It's 9:35.
Oh, okay. Yeah. I'm gonna skip over this. There's basically, you can also do protein binding. It's a pretty similar concept to motif scaffolding. I think the major thing to note here is that they were basically, they chose 5 protein targets and kind of did this experiment of protein binding, and they were able to yield like an overall 19% success rate in terms of successfully inhibiting the function of the target. And if we think back to like our original example from the like protein engineering pipeline, which is what we wanted to do, to be able to go through that computational pipeline and get a positive result and like 1 of every 5 designs is actually like a really good accuracy and as far as computational protein design goes. And so some key summary of the performance. So half of the design proteins show high expression, solubility, and stability, which is really good. And designs are novel, as we saw from that graph showing the comparison to PDB structures. And it's also highly like generalizable for conditional generation. So I went through like 3 or 4 examples of various conditional generation tasks, and it actually didn't require that much modification, right? The general framework is still very similar, just maybe you're changing some of the inputs or changing some of the priors. It can also design complex structures with long sequences. It's faster than existing methods, and it's like the other Baker Lab paper, it's very highly backed by experiments. So one example I think that's really cool is like they basically just casually came up with like some viable cancer therapeutics as like a side quest during this paper as like a experimental validation, which I think is really cool. And it just kind of shows how powerful this framework can be. Some limitations of RFDiffusion is so a lot of the new diffusion models actually are able to predict all atoms, and there's like de novo design, but RFDiffusion does just backbone. And that's definitely limiting, because what that also means is it requires some form of inverse folding to get the sequence. Whereas if you're doing all atom prediction, you know the atoms, you know the side chains, so you know the actual amino acids. So that's definitely a limitation of RFDiffusion. And then it's this version is limited to protein-only interactions, so it can't really model things like interactions with DNA and RNA or other like ligands. So yeah, just to kind of recap then, we've completed the computational part of a modern protein design paradigm that involves generating protein backbones with RFDiffusion. Typically, we want that to be conditioned on some specific desirable criteria. And then one can determine the sequence that would fold to make this backbone through ProteinMPNN. And lastly, the structure can be computationally verified through an AlphaFold prediction of the structure of the folded ProteinMPNN chain. And then the actual last step, and probably the most important, but in my opinion, maybe not the most exciting, I don't know if I want to be pipetting, but is the experimental validation. And I kid, but it's actually the most important part of this step because the computational results don't mean anything if we don't have like real life data backing it. And yeah, without that, none of this cool machine learning really means anything. So I have a couple more slides about like kind of next steps, but do we want to maybe
You go for it. Go for it. Yeah.
Okay. So yeah, I'll go fairly quickly through this. I think this is like pretty important actually. So think about like so structure prediction right now, AlphaFold methodology is kind of still the king. As mentioned, like basically they dominated all Cas15 entries, even though they literally weren't even in Cas15. And in 2022, they actually released a multimer version of the model, which addressed one of the main limitations I actually mentioned. And as of a couple of months ago, DeepMind announced that they were back in the kitchen cooking up a new AlphaFold that can deal with a larger variety of structure design problems, including like ligand and DNA binding, and even post-translational modifications. So that should be really exciting. And then lastly, if you recall earlier, like this idea of the two schools of thought with structure prediction, it definitely seems like one of the major next steps would be being able to integrate physics-based methods, because as I mentioned right now, like AlphaFold, because it's mostly based on evolutionary information, can't really deal with de novo mutations because it's not really reasoning through like physical means. So being able to at least integrate that to some extent, I think will definitely help improve predictions, and it'll also allow us to deal with cases where maybe we don't have homology information. Okay, so inverse folding, there have been some recent models, a few of which are like PyFold, which is a graph neural network, GradeIF, which actually use diffusion for inverse folding, and I think I found BiProt, which is like a language model that have achieved incremental accuracy improvements of just a few points over MPNN. But crucially, none of these methods are actually designed to deal with multiple chains. There's definitely one of ProteinMPNN's major advantages. It also seems to still be the best in terms of the accuracy of its AlphaFold predicted structures, like I kind of mentioned, in terms of like helping improve like stability of designed structures. There's actually also been pretty impressive improvements in antibody inverse folding. So there's specifically a method called AntiFold, and they actually just basically took a model that on its own performs worse than ProteinMPNN normally. It's called ESMIF, but they then fine-tuned it on antibody data sets, and they made a few other like minor tweaks, and it basically allowed them to like double sequence recovery accuracy. And it kind of makes sense because antibodies are have a very like specific like Y-shaped structure, and then limiting the types of protein that the model is working with makes it it only has to reason about this specific type of protein, so it's able to make better predictions. So that's definitely, I guess, in a sense, so that model is definitely performing better in accuracy on that specific task, but it's not as generalizable as ProteinMPNN. I will say it doesn't seem like there's too much hype around inverse folding right now, and that kind of actually has to do with the reasoning has to do with some of the new advancements in the backbone design space. And from my viewpoint, it seems like this is where a lot of the hype is right now. A lot of new models are related to this de novo backbone design. There've been a lot of new and interesting approaches, mostly based on a lot of them are based on diffusion, language models, or even some kind of combination. So there's ideas of like using natural language for protein design, similar to how you can maybe enter a prompt into GPT-4 or DALL-E, and it would actually just like create a protein, which is super cool. And it seems like an increasing number of these are focusing not just on backbone, but on this idea of like sequence structured code design, and actually trying to model all of the atoms in the protein rather than just the backbone. And so, as I mentioned, when we have all the atoms already in the de novo design. We don't really need inverse folding to be able to predict the sequence. Like we already have that information. That's part of the reason it seems like maybe, you know, inverse folding is not going to be as practical if it turns out that these all atom methods are become the new standard for de novo backbone design. So the other thing to note is that RF diffusion does kind of seem like it's still a top approach, but it's a little hard to exactly say because there's no great like standard benchmark set. Also, I think to really be able to benchmark this, you need a lot of experimental validation, and not a lot of labs do the same level of experimental validation as the Baker lab does. So as of right now, it does seem like RF diffusion is still on top, but I think that there seems to be a lot of progress in this area, and I think that'll probably change soon. And lastly, it seems that overall, and this makes sense, there's going to be a greater emphasis on conditional generation methods, because, you know, unconditional generation can be cool, but it's not really going to be practical for anything. And so, yeah, I think that concludes things. I've included a few like resources here. The top 3 are just like the models I talked about. There's a really good de novo protein design review from a couple of months ago. And there's also this, I found this GitHub of papers for like the topic of this talk, like protein design using deep learning. There are a lot of really great like benchmarking here, just like new papers and like reviews that I think have a lot of good information if you want to learn more. So yeah, thank you guys so much. Any last minute questions? Oh, yes.
Is AlphaFold to kind of use as like a pseudo round trip?
That's a really good question. I think, yeah, to some extent it kind of is. And it's because it's, as I mentioned, like the predictions it makes are really good. But I will say that if you look at the training data for all of these models, like, you know, ProteinMPNN and RF diffusion, they still only really used like experimentally validated structures. Because I don't think, like, I wouldn't say it's like good enough to where you would really want to be training high accuracy models on. But it's more so, I think, used for later steps in like kind of the benchmarking process. But yeah, it is. That's a good point. It is kind of used as like a pseudo ground truth. Yeah.
Wait, sorry, just follow. So like you said that in RF diffusion, those are like symmetry stuff.
Yeah.
Protein, big ones. But you also said that for AlphaFold too, it doesn't like estimate, it doesn't predict the structures of like those chain large proteins as well. But I'm guessing the RF diffusion doesn't really use AlphaFold 2 as a benchmark for like those kind of proteins, right?
Yeah.
Okay.
There are also, I can't remember off the top of my head, there might be some cases where they do use AlphaFold benchmarking, but they would probably be using the multimer version. So what I mentioned was like the base version. There is also like an AlphaFold multimer, so you theoretically could benchmark using that new AlphaFold version. Yeah.
Okay.
Terry?
I wanted to hear your insights about like data set versus model. I think we had a previous discussion when we were doing combination. Do you think if we should, we would be focusing on creating data set and then still really have a mature model, or do you think since we have a limited data set, we should be still focusing on the model architecture and try to confront that issue?
Yeah, I mean, I think that like I think always in machine learning, like having good data is like an underappreciated like kind of thing to have. I think that like, yeah, definitely having good data is like very important, but I think you can also, there definitely can still be improvements made in model architectures. I think in terms of like structure prediction, I think like the current like AlphaFold kind of paradigm, the architecture I think is really good. I'm curious to see how they're gonna like improve on it in the new version. But I think that in terms of structure prediction, probably the next steps would maybe just be trying to integrate some kind of physics-based reasoning. In terms of the other models, there definitely seems to be like, especially like inverse folding, there definitely seems to be room to improve, like just based on like sequence recovery metrics. So I feel like maybe the move there would be some kind of architecture change, but Obviously, like high quality data is always a plus. Yeah.
In the AlphaFold section of the presentation, we were talking about the pairing. You said that triangle inequality was kind of how it was used to determine popularity. What if you use like eigenrepresentation or other algorithms? Does that break the physics of the protein structure?
Sorry, what other algorithms?
Like eigenvectors like what Google uses in PageRank, like
Oh, okay.
other ways of finding popular nodes in a directed graph.
Uh,
Like, would that break like the physics behind proteins or whatnot, or? Is this just
honestly, that's a good question. I'm not too sure. I'm not too familiar with that algorithm. So I'm not too sure, but I imagine that I will say that like it definitely seems that the creators of AlphaFold put a lot of effort into like trying like every possible like optimization they could. And chances are if it's like an algorithm already used in Google, there's a good chance they thought about it. And maybe they even implemented it, but realized it didn't make sense or it didn't work out. But yeah, I'm not too sure.
But in general, like you talked about the triangle inequality.
Yeah.
Because it's like physical distances.
Yeah.
So that's
Yeah. The basic idea is they wanted to be able to reason about this, about the fact that like there's going to be there's not just two points connected to each other. There's like it's connected into this like geometrical space. So you can't just have this kind of contradiction of like 2 points being connected, like 3 points being connected in a line, but then the other like the 2 points on the edge just being like really far away from each other, because that violates the inequality.
Awesome.
Yeah.
Thank you so much.
Thank you guys.